Hi, everyone. Good morning. So just a couple quick things. Monday is our midterm. And please check BSpace for your room number. Just be sure that you're there before 8 o'clock in the correct room on Monday for the exam. Also, as another reminder, I'm holding a question and answer review session tonight. So if you want to come to that, it's going to be held at 155 Dwinell Hall, that big lecture hall, 7 to 9 p.m. tonight. Okay, so bring your questions, and it'll just be a strictly question and answer session tonight. So today what we're going to do is finish up talking about photosynthesis, chapter 10. And what I want to do is to talk about photosystems. We're going to talk about the Calvin cycle, so how the light reactions couple to basically provide the energy that drives the synthesis of organic molecules. And then we're going to talk at the end a little bit about adaptations that different kinds of plants have made to survive in environmental conditions that might otherwise not be so favorable for the photosynthetic reactions. So remember that last time we were talking about the excitation of chlorophyll molecules. So chlorophyll is basically, it's a pigment, right? It's a dye. We looked at the chemical structure of this, and there's not just one type of chlorophyll. There's multiple types. They vary slightly in their chemical properties. And the other thing to note is that the same type of chlorophyll molecule can actually be excited by different wavelengths of light, depending on the environment that it finds itself in inside of these, what we're going to talk about today, these large photosystems. But the principle is that a photon excites a chlorophyll molecule, and that basically knocks an electron from the ground state into an excited state. And this is, of course, very unstable. Decay happens very quickly. And if this is happening in isolation, we can actually observe this decay as the release of both heat and a photon. And of course, when this decay happens, this is not a perfect transfer of energy, right? There's some loss of energy as heat. And so the photon that gets released is at a lower energy or a longer wavelength than what we get for this initial excitation. And so we can actually observe this. If we have a purified solution of chlorophyll molecules, these molecules will actually fluoresce in the presence of light through this process. Now, of course, that doesn't happen in a plant, right? In a plant, the chlorophyll is it's embedded in the thylakoid membrane. It's part of these photosystems so that plants can capture that energy that when these excited electrons decay back to the ground state, that energy is not lost, but in fact it's captured and then used to drive ultimately the synthesis of organic molecules. So that's what we're talking about today, is how that actually works. It turns out that these pigments are contained within very large complexes called photosystems. These are complexes that have multiple proteins, they have lipids, they have many different types of pigments within them, and they're embedded within the thylakoid membrane. So these are complexes that include a central reaction center that is surrounded by what we call light harvesting complexes. And these light harvesting complexes are pigment molecules that are anchored in proteins within the light harvesting, within the photosystem itself. And the job of these pigments is to funnel energy of photons that are interacting with it into the reaction center. So they're a way of capturing electromagnetic radiation that is interacting with the photosystem and directing it to the place at the center where chemistry can occur. And there's a primary electron acceptor in the reaction center whose job is to accept an excited electron from chlorophyll A. These are pigments that are embedded within the photosystem, and as we're going to see, there's multiple copies of chlorophyll A, but two of them are special. They're sort of located adjacent to the primary electron acceptor, and their job is to transfer that energy after excitation of an electron by interaction with a photon of a very specific wavelength. And really what this is the solar-powered transfer of an electron from chlorophyll A molecule, to the primary electron acceptor. This is the central first step of the light reactions in photosynthesis. And I wanted to show you first a crystal structure of photosystem 2. So there have been a lot of experiments done to try to understand the structure of these photosystems. They're incredibly complex, literally complex complexes. They have multiple components, and they're really beautifully set up in the membrane to capture energy that is coming in the form of light and transferring that in a way that's useful to the plant. This shows you a picture of a crystal structure of one of these photosystem 2 complexes. And I think you can tell by looking at this that this is first of all, it's very big. In this particular example, there are 20 different polypeptides in this complex. And so what you're seeing here you can see all these alpha helical elements. So these are all the membrane spanning parts of the protein, and those are all in gray. So those are all the protein parts of this complex. And then you can see there's some additional protein parts that are down here. Other proteins that are associated with the complex but are not actually embedded within the membrane are located down here. We have 35 different chlorophyll A molecules. So your book shows you that cartoon that has, we'll look at that in a moment, that has the chlorophyll A pair located in that reaction center. But remember that there's a lot of additional chlorophylls as well that are in this complex. And in addition to those additional chlorophylls, we also have other pigments, carotenoids, that are located in this complex. We talked about those a little bit last time. Their job is to, in part at least, is to deal with excess energy that would otherwise be damaging to the molecules in these complexes, right? So their job is to help protect the integrity of these structures. And then finally, there's also quite a large number of integral lipids. And we can see those here in these yellow and red colors. These are lipids that are actually embedded within the complex, and their job is to help anchor this within the lipid bilayer of the membrane. So very large set of molecules that come together in these photosystems. And if you think about it, it's actually quite an amazing achievement that it's been possible to crystallize these things. And so a lot of effort went in over many years in a number of laboratories to find ways to actually purify these complexes in a way that they were fairly homogeneous, and they could actually be crystallized, and X-ray diffraction could be used to eventually solve a molecular structure of this. So a lot of what we know about the way that these complexes work is based on having high-resolution structural information for how they're actually put together. And the other thing I wanted to just show you here in a cartoon is, so this is now just very cartoony version of what I showed you on the last slide in detail, which is the photosystem. And this is just pointing out that this thing is embedded in the bilayer, of course, and there's a directionality to it, as well as the presence of these different kinds of pigments. In this case, this is focusing on plastoquinones that are anchored in specific sites within these complexes through long hydrophobic tails that extend from the end of the pigment and extend out towards the lipid bilayer, the thylakoid membrane in this case. And this is just showing the chemical structure of this. These long isoprene tails that have multiple isoprene units on them that are very hydrophobic, and they actually help to anchor these pigments in the photosystem. So now let's look at, this is now a cartoon taken from the book that shows this structure much more schematically. And you can see that this is really situated very nicely in the lipid bilayer. And you know from that crystal structure that I showed you that the way that is anchored is through these alpha helices in the proteins that are transmembrane, right? They're going back and forth across the membrane, so that's what anchors this in the lipid bilayer. And then these are set up so that the pigment molecules are organized such that when a photon interacts with one of them, there's a very convenient transfer of energy that occurs that leads ultimately to, effectively, it's like fine-tuning. It's filtering the energy so that we get exactly the right wavelength that's appropriate to excite electrons in this central pair of chlorophyll molecules right here in the reaction center complex. And when that happens, we get transferred an electron to the primary electron acceptor here, and then that triggers a series of electron transport reactions that lead to, ultimately, to the production of a proton gradient and the synthesis of ATP, very analogous to what we saw in oxidative phosphorylation. Question? These are all going to be redox reactions, okay? So very similar to what we saw with oxidative phosphorylation. Yeah. So there, as we said, there are two types of, or maybe I didn't say this, but there are two types of photosystems in the thylakoid membrane. Photosystem 2 functions first. So that was the one that we just looked at the structure of. So this is, this is one of those things in biology where actually the discovery of these photosystems and their naming occurred in one order, but eventually when people figured out what they were actually doing, it turns out photosystem 2, and not photosystem 1, is actually the first photosystem involved in this set of reactions. So it's a little bit, just got to deal with that nomenclature there. So the photosystem 2 operates first, and it's actually fine-tuned to absorb a wavelength of light 680 nanometers. Okay, so this is the wavelength that will excite that pair of chlorophyll pigments in photosystem 2. And so we call the reaction center chlorophyll of photosystem 2 P680. Okay, P680. Yeah, question? So I think what's being asked here is what happens when photons initially interact with these light harvesting complexes. They're not obviously not all of those photons are, I mean, it's white light usually, right? So it's not all at 680 nanometers. So it comes in, and then there's a series of electron transfer reactions that occur that lead to eventually a energy that's fine-tuned to excite an electron at this wavelength. Does that make sense? It's like a filtering kind of thing. If that, yeah, ask me later. Okay. So. And then we have photosystem 1, which is fine-tuned to absorb light with a wavelength of 700 nanometers, so slightly different. And interestingly, when people have looked at the structure of photosystem 1, turns out chlorophyll A is also in the reaction center of photosystem 1, right? Same chemical structure. What's interesting there is that it's in a slightly different environment. So it actually the excitation energy is adjusted slightly, not by the actual chemistry of the chlorophyll molecule, which is the same, but by the chemical environment that it's in within the reaction center. Slightly different in these two photosystems. And so when this process is initiated, there are two different ways that electrons can actually be triggered to flow through the system. There's linear electron flow, and then there's cyclic electron flow. We'll talk about both of those, sort of how they work and what happens when they are triggered. So in linear electron flow, we have, as I said, we have these two possibilities. And in linear electron flow, which is the primary pathway, this is a pathway that involves both of these photosystems, photosystem 1 and photosystem 2. And this linear flow of electrons produces ATP and NADPH, and it uses light energy to do this. And so, just as we saw in oxidative phosphorylation, the synthesis of ATP is not a direct consequence of interaction of these photosystems with light, right? In fact, it's an indirect consequence because what we're going to see is that these electron transport pathways that get set up in response to or that are triggered by interaction with photons lead to the production of a proton gradient. And once again, we have just very analogous to what we saw in oxidative phosphorylation. We have a proton gradient that will end up driving ATP synthase by chemiosmosis. In other words, the high concentration of protons on one side of the membrane traveling down its concentration gradient through these big ATP synthase complexes that we looked at in the previous lecture and driving ATP synthesis, very similar to what we've seen in mitochondria. So let's just walk through this linear flow of electron pathway. So what happens to kick this all off is that a photon hits a pigment molecule, and its energy is passed among different pigment molecules until eventually there's the excitation of P680 in the reaction center right here. And so this is just showing you incident light coming in. There's a transfer of energy to these different pigment molecules, eventually leading to excitation of P680 in the reaction center and transfer of an electron to the primary acceptor right here. And when P680 receives interacts with light at 680 nanometers, it basically loses an electron and becomes P680 plus. This is a very strong oxidizing agent. In fact, it's thought to be certainly one of the most, if not the most, the strongest oxidizing agents known in biology. It's very strong, and this basically drives the splitting of water that's catalyzed by enzymes that are in this reaction center, and electrons are transferred from the hydrogen atoms back to P680 plus, and that reduces it back to P680. And in the process, protons are released into the thylakoid lumen along with oxygen. And this, of course, oxygen is released as a byproduct in this reaction. So it's really kind of a waste product, but it's really the primary driver of the concentration of oxygen in the Earth's atmosphere. Are these the same photons? Same protons that will be used in the gradient, that's right. So these are starting to build up a proton gradient that will be used to drive ATP synthesis later on. And then, so once that initial electron transfer reaction occurs, then there's a series of transfers as each electron essentially falls down an electron transport chain, starting with the primary electron acceptor in photosystem 2 and then over to photosystem 1. This is a very schematic drawing. It's obviously not showing you all the detailed reactions here, but the idea is that there's a transfer to plastoquinone, to cytochrome complex, and then ultimately to plastocyanin. So these are all pigments that are capable of redox reactions where we have transport of electrons along this chain from starting at a higher energy level and ending up at a lower energy level. And energy that's released by this fall drives the creation of a proton gradient. So in addition to protons produced here, we also have protons that are being produced here and helping to drive the creation of this gradient across the thylakoid membrane. And then it's the diffusion of those protons back across the membrane along their concentration gradient that drives ATP synthesis. And this is showing the where this ends up. So we have this electron transport chain. And then in photosystem 1, which is over here, and again, photosystem 1 is another one of these very large complexes with multiple protein, multiple polypeptides, multiple pigment molecules, et cetera, in the complex. And at the center, we have this, again, this pair of chlorophyll molecules that are finely tuned to be excited at a wavelength of 700 nanometers. So this is once the transferred light energy excites this pair of chlorophylls, which lose an electron to another primary acceptor up here, again, we have then a oxidized P700, P700 plus missing electron that accepts an electron that's passed down from photosystem 2 through this electron transport chain. So there's a couple things going on here, right? One is that we've got this electrons being transported along this chain. We also have light that can interact with receptor molecules in this complex as well and can lead to excitation here. Okay, so two ways. And each electron falls down this electron transport chain from the primary electron acceptor. And in this case, if we're going from photosystem 1, it ends up in a protein called ferredoxin. And in the process of this set of electron transfer reactions, there's no proton gradient that's created. So in going from photosystem 1 reaction center to ferredoxin, there's actually no gradient that's created. So there's no ATP that results directly from that set of electron transfers, not like going from photosystem 2 to photosystem 1. These electrons are then transferred ultimately to NADP plus and reduce it to NADPH. And again, analogous to what we saw in oxidative phosphorylation, this is another storage molecule. So the electrons of the reduced form, this NADPH, are now available for the reactions of the Calvin cycle, which is where synthesis of organic molecules actually occurs. So you can see that there's two ways, right, that the system is able to harvest energy from incoming photons. One is by creating a proton gradient through these electron transfer reactions, and the other is by generating NADPH, which can then be used to help drive the Calvin cycle. And this is just showing those downstream steps. After photosystem 1, we have this electron transfer reaction here, and then transfer to ferredoxin, this electron transport chain that doesn't lead to any proton gradient being created, but does eventually, at the end, lead to reduction of NADP plus to NADPH. And of course, that's catalyzed by an enzyme, NADP reductase, that produces NADPH at the end of this linear chain of reactions. Question? She's asking, why do we even need photosystem 2? Because here we can also capture light, right? And it turns out some organisms actually don't have a photosystem 2, right? Some of them don't. But you actually get a lot more if you do have photosystem 2 in terms of the ability to harvest energy. And the reason is that if you can't start off over here, then you don't have the ability to generate the proton gradient through this set of transfer reactions or through the splitting of water over here. No, from photosystem 1. Well, where does it get its source of replenishing electrons? I mean, this is, of course, an aqueous environment. There's water around, and but it's just not efficient the way it is if you start off over here. And that actually plays into the next thing that I want to mention, which is that in addition to a linear flow of electrons, we can also have a cyclic flow of electrons. And this is a situation where this happens in plants that have both, or organisms that have both photosystems, but also in organisms that have just photosystem 1, in which we see that we get the production of ATP, but not NADPH. And so this is showing you how that cyclic flow actually works, where we have, this is, in the background is the pathway that we just looked at, and this is showing you that in the case where we have a cyclic flow of electrons, instead of electron transfer reactions going in this direction, where we have the transfer to the primary acceptor in photosystem 1 and then transfer to ferredoxin and ultimately reduction of NADP plus to NADPH, instead what happens is that we get transferred to ferredoxin, right, and this can even be the same ferredoxin, but instead of going on down this chain and leading to ultimately to the reduction of NADP plus, instead what happens is that it feeds back into this pathway over here, where we get electron transfer reactions through the cytochrome complex and down to plastocyanin, and then feeding back into photosystem 1. And so when this happens, this is a pathway that will generate surplus ATP, because we can, by going through this pathway, we actually can still trigger ATP synthesis through creating a proton gradient, but we actually don't build up NADPH. And the thinking here is that this is a way that plants can actually siphon off some of the energy that's coming through this pathway and use it to generate, build up even more ATP by doing this, running this cyclic flow of electrons over here. So again, the proton gradient is generated over here, right, and not over here. And that's why it's showing you that ATP results from this pathway, but not directly from this pathway. And so if we compare what's going on in chemiosmosis in chloroplasts and mitochondria, they can generate ATP in both cases, but they're using different sources of energy, right? So mitochondria transfer chemical energy from food molecules, organic molecules, to ATP, and chloroplasts, in contrast, are transforming light energy into the chemical energy of ATP. And then if you look at, I have a slide of this in a couple of slides, showing the spatial organization of chemiosmosis. It's a bit different between chloroplasts and mitochondria, but in both cases, what we see is that these electron transfer reactions are occurring in the membrane, and so that the proton gradient can be established across that membrane and then be used to drive ATP synthase and the production of ATP. So in mitochondria, we have protons that are pumped into the intermembrane space, where they drive ATP synthesis as they diffuse back into the mitochondrial matrix, whereas in chloroplasts, protons are pumped into the thylakoid space, and they drive ATP synthesis as they diffuse back into the stroma. So, yeah, question? So she's asking, where are the chlorophyll B and carotenoid molecules functioning? So, just going back to, if you look at a cartoon, this is kind of a very cartoony picture, so there would be some of these pigment molecules in here, and if you look at the actual structure here, they would be embedded within this large complex. And so again, their job is to be available to interact with photons that come in, or with energy that gets transferred after photons have interacted with initial pigment molecules. And so there's this very efficient transfer of energy between these different pigments. And they're, depending on their environments and their chemical